Today we will discuss conjugation in bacteria. Bacteria are prokaryotic organisms which do not reproduce by true sexual means. True sexual means through the production of haploid gametes followed by their fusion to produce a diploid cell, right? But bacteria have a unique way of sexual reproduction where the genetic material in some form is exchanged between the donor and the recipient cell, right? So sexual reproduction in bacteria is of three types: conjugation, transformation, and transduction. So today we will discuss only conjugation in detail. Some bacteria are capable of transferring pieces of their genes to other bacteria by coming close to each other, right? They means bacteria come close to each other with the help of pili. Pili, hair-like structure present on the surface of the bacteria, right? They help in this process. When they come close to each other, they get interconnected with the help of a conjugation bridge, right? This process usually involves a donor strain and a recipient strain. Donor is often called F plus strain because it has F plasmid, while the recipient is called F minus strain because it is devoid of, means it lacks F plasmid or fertility factor. F plasmid is also known as fertility factor, right? So by the end of this method, F negative is also converted into F positive, means recipient is also converted into donor, and this process is called complete conjugation, right? How this conjugation process was discovered? It was discovered in 1946 by Joshua Lederberg and Edward Tatum. How they discovered this process? In the laboratory, they took two strains of Escherichia coli with different nutritional requirement, strain A and strain B. Strain A would grow on a minimal medium only if the medium were supplemented with methionine and biotin strain, right? Methionine is an amino acid and it is required for protein synthesis. Similarly, biotin, it is associated with lipid synthesis, and biotin is critical for bacterial survival and pathogenesis, right? Strain B would grow on a minimal medium only if it were supplemented with threonine, leucine, and thiamine amino acids, right? Lederberg and Tatum plated some of the dishes only with strain A bacteria. You can see here only strain A, and some only with strain B bacteria, and some with mixture of strain A and B both, that had been incubated together for several hours in this liquid medium containing all the supplements, right? What they observed, no colonies arose on plates containing either strain A or strain B alone, showing that there was no back mutation which restored the ability to grow on unsupplemented minimal medium, right? However, the plates that received the mixture of both the strains produced colonies. You can see this blue colored, right? So this observation suggested that some form of recombination of genes was there between the genomes of two strains to produce prototrophs. Now, what are prototrophs? Prototrophs are self-sufficient producers of all the required metabolites. In other words, we can say wild type, right? Conjugation is initiated by a signal when relaxase enzyme creates a nick in one strand of the conjugative plasmid at the ORI-T region. Now, what is this plasmid, ORI-T and all, right? So before starting with the conjugation process, we will discuss first what is this plasmid, so or F plasmid. F plasmid is an episome. Episome means a plasmid that can integrate itself into bacterial chromosome by homologous recombination. So this episome uh, with a length of about 100 kilobase pair, it carries its own region of replication, ORI-V, and an region of transfer, ORI-T region. I will show this region and structure of plasmid. Then there can only be one copy of F plasmid in a given bacterium, either free or integrated. And the bacteria that possess a copy called F positive bacteria, or and it is designated as F plus. Cell that lack uh, F plasmid are called F negative or F minus, right? F positive bacteria function as donor and F negative function as recipient cell, right? So you can see here the structure of plasmid, here ORI-T region, right? Here. Among other genetic information of F plasmid, it carries TRA and TRA region and TRB locus, which together is about 33 kilobase pair long and consist of 40 genes, this region, right? TRA locus include the pili gene and regulatory genes, which together form pili on the surface of the cell. You know, pili are the small hair-like outgrowth or hair-like structure present on the surface of bacterial cell, right? The locus also include the genes for the proteins that attach themselves to the surface of F negative bacteria and initiate conjugation, right? Now, steps of conjugation. As I told you, conjugation is initiated by a signal when relaxase enzyme creates a nick. You can just see here, donor cell and recipient cell. Donor cell has plasmid, while in recipient cell no plasmid is there. So relaxase enzyme creates a nick in one of the strands of this conjugative plasmid at the ORI-T region, right? Relaxase enzyme may work alone or in a complex with over a dozen of proteins. Complex of relaxase and protein is called relaxosome. F plasmid system, when the relaxase enzyme, it is called TRAL, and in as a relaxosome it consists of TRAL, TRAY, TRAM, and the integrated host factor IHF, right? When look at this second diagram, then this nicked strand or T strand then unwound first from the unbroken strand and then transferred to the recipient cell in a 5' terminus to 3' terminus direction, right? So 5' N means has a free hydroxyl or phosphate group on fifth carbon, while in the case of 3' end, it has a free hydroxyl or phosphate group on third carbon, right? Remaining strand is replicated. You can see here, right? Replicated either independent of conjugative action or in concert with conjugation. So conjugative replication may require second nick before the successful transfer can occur, right? So you can see here in the second diagram, pili of the donor cell. This is donor cell having plasmid and this is recipient cell. Draw the recipient closer. This is pili, pilus, sorry, singular, right? Allowing the fDNA, this DNA, now look at this diagram, pass through the pore of the recipient cell, right? So one strand of double-stranded fDNA is transferred to then DNA uh, transferred, and the DNA replication restores the complementary strand in both the donor and the recipient, right? So E. coli, uh, in the case of E. coli, transfer of entire bacterial chromosome takes about 100 minutes. Now, formation of the high frequency recombination strain. Now, what is this high frequency recombination strain? This is another strain of bacteria discovered by Luca Cavalli Sforza in 1952. When he crossed this high frequency strain with F negative strain, high frequency strain produced 1,000 times more recombinants than normal F positive strain. So Cavalli Sforza designated this as high frequency strain, right? So during conjugation between high frequency cell and F negative cell, a part of the chromosome is transferred, then it sometime due to random breakage, it interrupts the transfer before the entire chromosome is transferred, right? Here, this is a high frequency strain, genome and plasmid. This is when here the conjugation bridge is there, pilus, helping the bacteria to coming close to each other. Then a part of the bacterial, donor bacterial DNA transferred to the recipient cell, but sometimes random breakage interrupt the transfer before the entire chromosome is transferred, right? You can see here in this diagram. Then chromosomal fragment can then recombine with the recipient chromosome here, right? So this result in the transfer of selectable marker from the high frequency donor to recipient cell. So transferred strand is replicated in the recipient cell and the donor genes may become incorporated in the recipient chromosome through crossover, right? So at a later stage, transferred fragments of DNA in the recipient are lost in the course of cell division. So here in this process, recipient cell remains recipient, will not transferred to donor cells, right? What are the applications of conjugation in bacteria? First, transfer of genes from bacteria to yeast. Also in the case of plants, even to mammalian mitochondria. Conjugation has advantage over other forms of genetic transfer, including minimal disruption of the target's cellular envelope and the ability to transfer relatively large amount of genetic material. And third, in plant engendering via TI plasmid, right? So this is all about conjugation in bacteria. Thank you.